filtration, this is where we have return of filtered molecules to the blood, and so 85% of reabsorption occurs in the proximal tubule and the descending loop of Henle for the nephron. Okay, so with the filtration, this must leave the tubular fluid by crossing the luminal membrane of the tubular cells. All right, and so when we look at this image here, we have the filtrate, and it's going to pass then through the tubular epithelium, and then it has to pass into the interstitial fluid that's between the tubule and the vessels, and then finally enter into the peritubular capillary. So all in all then, we're looking at 5 barriers for these substances to be reabsorbed into, and so this is also very selective and variable process to really keep in only the ions that we and molecules that our body actually does need. With this process, then we have both passive and active reabsorption that occur. And as a reminder, the passive reabsorption, this means that there's no energy required, okay, for movement. And so this is going to occur down one's electrochemical or osmotic gradient. So the ions and substances that this happens for, chloride, urea, and then water by osmosis. Okay, active reabsorption then. This is where now we're going to have energy. Hopefully a little bit of a reminder. All right, so this is going to go against its electrochemical gradient, and so this occurs for sodium, glucose, amino acids, and then different organic substances. And so for these cells that have a lower sodium concentration than the filtrate inside the tubule, this is due to the sodium-potassium pumps on the basal side of the cells and also a low permeability to sodium. So then sodium from the filtrate will diffuse into these cells and then is pumped to the other side due to that protein pump. So if you remember, a lot of ATP is needed for sodium and potassium to pump sodium back into the cell. All right, so there is though a maximum though that we have. So this is the transport maximum that has a limited amount of materials that can be transported. So once we are saturated with this, that means that no more of these ions can be pumped back in. And so these are going to not be reabsorbed then, and then they will be just excreted from the body. One of the most important ions to be reabsorbed is sodium. And again, this is due to an active process, and the sodium ATPase pump will be responsible for that, okay? So this is essential for sodium reabsorption. Majority of where sodium is reabsorbed is going to be within the proximal tubule, so about 67%, and this is important because this helps to also reabsorb glucose, amino acids, water, chloride, and urea. So as sodium is being reabsorbed, then it's also reabsorbing these ions and molecules. Now the other important thing though with the proximal tubule is that, I'm going to use different color now, with water. So the same percentage of water is going to be reabsorbed as that of sodium. So they will match each other, and we'll get to that in the next slide, okay? All right, so then another 25% is going to be reabsorbed within the ascending limb. And the important thing here, though, is to remember is that the ascending limb then is going to be impermeable to water. So this is going to help with the concentration of urine, okay? And different hormones are going to affect then how much is going to be reabsorbed and for that concentration. So then, since we have an impermeability to water for the ascending limb, this means that sodium is impermeable in the descending limb, okay, for that loop of Henle. And then the last area that we have are the distal and the collecting tubules. So here we have the last percentage of sodium as well as for water that's going to be reabsorbed. Okay, so here we'll have a last bit of hormonal control for the reabsorption, and so this helps to regulate the extracellular fluid volume for our cells. And so, again, key thing, sodium is going to be reabsorbed, active process, and to show you just how important this is, here, this figure right here, the stat, 80% of what we use for energy of the kidneys is spent to reabsorb that sodium. So highly important to conserve and keep as much of that sodium as possible. So we just covered in the last slide about the impermeability differences to those different ions. Okay, so this is termed the concurrent multiplier system. So this has to do with NaCl and with water. Okay, and so within the descending limb, what we have here is that this is permeable to water. Okay, so water is going to come out and enter into the capillaries. So what we see is that the osmolarity will change. So we're going from 300 milliosmoles down or up to 1200 milliosmoles. This means as water is leaving, we're becoming more concentrated. Okay. And so again, this is happening at the descending limb of that loop of Henle for that tubular component. All right, so now in the ascending limb, now water is impermeable, and now we have chloride ions that are going to flow out, okay? So now our osmolarity will change. So we're going from this highly concentrated, this 1200 milliosmoles, and now we are going down to 100 milliosmoles. Okay, so this is now to match then the water that has left. All right, so now our urine is now becoming less concentrated. And so these opposing flow in the loop then are going to create these hyperosmotic solutes that are going to be determined then on a need-to-need basis of the body. Okay, so in situations where we have more water though, we're going to have more dilute urine, and in dehydrated states though, we'll have more concentrated urine. And that's due to then with dehydrated states, we're going to conserve all of the water, whereas with more dilute urine, we are not going to conserve as much. And so the chlorine ions can't match that of the excess of water. So the major determinant of the permeability and passive water reabsorption in the collecting ducts is due to ADH, antidiuretic hormone, or vasopressin, as we've seen this before. All right, so the purpose of when we have ADH present, then this helps to stimulate insertion of aquaporins. And so these aquaporins then are going to be found within the collecting duct of the membrane there. And so this allows an increase in diffusion of water down its concentration gradient. Okay, so this high concentration of ADH then is going to increase the permeability of water in the collecting ducts, and so water is going to be reabsorbed maximally, and so we'll see a very small urine output. When we have no ADH present, this is when there are no insertion of extra aquaporins, and so this is going to increase then the amount of urine. Okay, so can you guess which one would be present when we have a dehydrated state? So that would be when we have then an increase in ADH, so similar to that of panel A. Good. All right, and then one thing also to mention here is that we have an obligatory loss of water, and this is equal to 400 milliliters per day. So even if you are stuck in the desert, have been dehydrated for, I don't know, a week, which hopefully never happens to you, but no matter what, based on cellular respiration and tissue metabolism, our body is going to excrete 400 milliliters each day. And again, this is just an obligatory that we have to get rid of as waste product from cellular metabolism mostly. Another important molecule to mention is of urea and how it helps to establish a hypertonic interstitium for the medullary. And so urea, this is going to be the waste product of protein metabolism, and the main purpose that it has is to work with a concurrent multiplier system of sodium and of water and help to establish the osmolarity differences. And so if we look at this graph here, or I'm sorry, this figure here, this yellow denotes the pathway of urea and how it is continuously reabsorbed, so that we have about 45 to 55% that is going to be reabsorbed. And so here, if you notice within the loop of Henle, this is where it plays its role in helping with the osmolarity changes within sodium and of chlorine. Okay, and the last role that it has too is here within the collecting duct, where under hormonal influences will impact then the amount of water that's going to be permeable. And so urea is also a mechanism in which helps with the concentration gradient for water and helps to regulate that even without hormonal influences that are needed. All right, as I said before, let's be very familiar with the RAS system. And so renin, angiotensin, aldosterone, we talked about this back in chapter 14, I believe, cardiac output. And so with this is going to be the most important one for regulating sodium, okay? And so this mechanism has been studied by physiologists for many, many years, and so we understand the system the best. All right, so we're going to talk about the hormones, and then the next slide I have a visual so that we can go through this. But let's start with aldosterone. And so aldosterone, the stimulus here, what I have is that we have a decrease in volume of blood. Also, we have an increase in potassium ion concentration or an increase in sympathetic activity. Then will influence aldosterone secretion. And so the effect then for the decrease in blood volume will be to reabsorb sodium and then the distal tubule and the collecting ducts. So with increase in potassium, we'll have an increase in potassium secretion in the distal tubule, so that exits the body. And then an increase in sympathetic activity is going to constrict arterioles. Renin, now this is going to be stimulated by a decrease in plasma sodium levels, so where this would be mostly triggered by blood volume. And so then the renin is going, the effect then is to convert angiotensinogen to angiotensin 1. So this is going to start that process. So ACE, the angiotensin converting enzyme, is going to be stimulated too when we have a decrease in plasma sodium levels, and then this converts angiotensin 1 to angiotensin 2. And then lastly, we'll have angiotensin 2 is going to be detecting low sodium levels, and so this is going to stimulate aldosterone. All right, so looking at this then, what we have is when we have a decrease in sodium, a decrease in extracellular fluid volume, or we have a decrease in arterial blood pressure, then we're going to have then the conversion of renin be stimulating with the enzyme, and so angiotensinogen is going to be converted into angiotensin 1, and so the kidneys are ultimately what will detect it. And so again, the liver is what houses the angiotensinogen to enter the bloodstream, and then the enzyme will activate it to angiotensin 1. Then the lungs are going to secrete the ACE, angiotensin converting enzyme, so we go from angiotensin 1 to angiotensin 2. And then the adrenal cortex then is going to be stimulated, where it's going to produce an increase in aldosterone. And so angiotensin 2 then will have the effects of increasing thirst, increasing vasopressin, and also to vasoconstrict. And so with the vasopressin, what this does, this is going to increase water reabsorption. Thirst is going to increase fluid intake, so this has to do with higher centers within the brain. And then the vasoconstriction then is going to cause vessels to constrict and then decreasing the blood flow until we get back to normal, and so we can get an increase in blood pressure. Now, when we have higher aldosterone levels, this is going to be stimulating within the kidneys, and so the kidneys then, within their tubular component, are going to increase sodium reabsorption and chloride will follow with sodium, okay? So sodium is going to be conserved, and then based on the concurrent multiplier system, then water is also going to be conserved, and so this will help to correct then to increase sodium salt levels, increase either our extracellular fluid volume, and also to help increase blood pressure. So this will continue on until we go back to normal homeostatic levels. All right, so this figure, we've seen this before when we talk about cardiovascular system. And so ANP, atrial natriuretic peptide, this is going to inhibit sodium reabsorption, so combating that of the RAS system. And this is going to be secreted by the atria of the heart in response to being stretched by sodium retention, okay? And so this will be due to then increasing blood pressure, and so that's going to all affect and activate ANP. All right, so then what happens is that we'll have stimulation of the kidneys to excrete more salt, okay? So decrease in sodium and water reabsorption. And because of the concurrent multiplier system, when we have a decrease in sodium that's being reabsorbed, water also is going to be decreased in reabsorption. This is going to decrease then the blood volume, and so ultimately we'll have a decrease in blood pressure. So now here, stimulus is an increase in blood pressure, whereas the RAS system is typically a decrease in blood pressure, as well as a decrease in the amount of sodium chloride ions. So this is like if you're on a high salt diet, you have a high blood pressure. So this are the mechanisms that are working within our kidneys and our bloodstream to help combat that.